Okay, so once again, try this again. Alright, we're going to go over DNA and genetics. We're going to start with inheritance, and then we're going to get into how DNA is replicated. And we'll get more into how DNA is turned into products, proteins, when we talk about DNA again next time. So, for many centuries, people have noticed that your parents and your children look similar, look somewhat like a blended combination of your mother and your father, yet they aren't all the same. So there's some sort of information that's being inherited from father to mother, or from parent to offspring. But the ideas weren't never really made concrete until Gregor Mendel, who is considered the father of genetics, and used pea plants to construct models for inheritance. Okay, the reason why he chose to use pea plants is because that you can produce pea hybrids. You can cross one pea plant with a different pea plant. And there were lots of pea varieties available, and they were quick and easy to grow. They're small. They didn't take a long time to mature. And they can self-fertilize, meaning you can take a flower and use that to fertilize itself, and a pea will grow. Or you can cross-fertilize, meaning you can take two flowers from different plants, and they will also make offspring. So some terms that you need to know before we get into Mendel's experiment is that there are true breeding individuals, and what happens when you self-fertilize a true breeding individual, all the progeny will have the same trait. So a purple flower that, when is self-fertilized, creates progeny that are also all purple. Okay, then you have, if you take two true breeding plants, so if you have one that is purple and one that is white, and you cross them, that's called a monohybrid cross. In this cross, you only have two variations. So we have purple or white flowers. They're both true breeding, meaning if they're self-fertilized, they will create only purple flowers or only white flowers. But when you cross them, that's when you now have a monohybrid cross. The first generation of a monohybrid cross is called the F1 generation. And then the F2 generation is you take members of the F1 generation and you have them self-fertilize. So what Mendel did was he took true breeding plants and did a monohybrid cross. He took true breeding purple and true breeding white, and he cross-fertilized them. And all of the F1 generation had one trait. So the purple flowers and the white flowers were bred, and only purple flowers showed up in the F1 generation. That was called the dominant, and then the recessive would have been the white. There were no intermediate characters, so there was no purple with white spots, there was no white with purple spots, there was no pink, no blue, nothing in between. It was just purple. And this was against the theory of blended inheritance, which is that there was just a combination of the two things. Okay, it was either purple or white. There was nothing in between. He didn't stop there, though. He also did an F2 generation, so he self-crossed those purple flowers, and then the recessive trait returned. Okay, the white flowers showed up in a 3-to-1 ratio. So 3 purples to 1 white in the F2 generation, even though when you self-crossed, they were purple. So what Gregor Mendel found out was that the 3-to-1 is actually a 1 to 2-to-1 ratio. Sorry, there's a 1 missing there. So the F2 plants, 3/4 of them were dominant, 3/4 of them were purple, 1/4 of them were white. But when he looked at them and he self-crossed these individual purple and white plants, he found that these two of the 4, were not true breeding. One of them was true breeding dominant, and the other one was true breeding recessive. So phenotypically, or the expression of the color was, yes, in a 3-to-1, but the genes within them were a 1 to 2-to-1 ratio. So Mendel came up with these 5 elements in his model, or his theory of inheritance. And the first one is that parents transmit a discrete factor. We call these genes now, but he just called them discrete factors. And each individual only contains 2 copies, one from their parent, each parent. And not all copies are identical. And an allele, then, is an alternative form of a gene. So we have purple allele and we have a white allele for flower color. If you have 2 of the same allele, you're homozygous. If you have 2 purple alleles, you're homozygous. If you have different alleles, if you have a purple and a white allele, then you're heterozygous. Fourth principle is that alleles remain discrete or unique. There's no blending. So if you get a purple, it's just going to be purple. It doesn't change to pink or blue or aquamarine allele. And because you have dominant and recessive alleles, just because you have an allele doesn't mean it's going to be expressed. If you have a recessive allele, but you have that white-purple flower allele, it's not going to be expressed if it's paired up with the dominant allele. And so then he started to construct a genotypic, or the alleles of an individual, which is called the genotype. And he separated that from the phenotype, which is the physical expression of a genotype. Okay, so you may be heterozygous, which means you have a big P. You have the gene for purple flowers, and you may have the gene for white flowers. Okay, and that's your genotype. But your phenotype would be purple. So what he determined based on these 5 principles is that genes segregate, and this is called the principle of segregation. So 2 alleles for a gene, here purple and white, during a gamete formation, or formation of an egg and sperm, are separated, and then they rejoin at random during fertilization. And we know this happens because we know what happens during meiosis. Gregor Mendel did not know this, but we know that the alleles on different chromosomes line up in the middle of the cell in random fashion, and then they come back during fertilization, and you get one from each parent. But they are not linked in any way, or at least not in these genes. So in order to show the genotype and phenotypes, we use what's called Punnett Squares. And we use letters to denote the dominant and recessive alleles. So a big P is dominant for purple, little p is white, which is the recessive allele. If you have a heterozygous individual and you pair those together with another heterozygote, then you get these different genotypes. A homozygous recessive would be little p, and that would be white true breeding. A big P would be homozygous dominant, and that would be a true breeding purple plant. And then you have a heterozygote, would be 1 purple and 1 white allele, and these are not true breeding. And there's your 1 to 2 to 1 ratio. There are many human traits that are dominant and recessive. For example, albinism, which causes white skin, white hair. A loss of pigment is a recessive phenotype. Whereas polydactyly, so having multiple fingers or more than 5 fingers, is a dominant trait that will be passed on if there is only 1 allele. When we look at these in pedigrees, we can determine the type of whether it's dominant or recessive based on the inheritance patterns of children and offspring. Okay, so this is a dominant and this is a recessive pedigree chart. But again, Mendel didn't stop there. He went on to do dihybrid crosses, which means that you have 2 separate traits. So we're looking at color of the pea and whether it's round or smooth, the texture of it. And when you look at them together, so if big R is for round, little r is for wrinkled. Big Y is for yellow, little y is for green. And what he did is he took two true breeding, round yellow and wrinkled green, and he crossed them. Again, he found only the dominant trait showed up in the F1 generation. When he allowed them to self-fertilize, though, he found a different ratio. It wasn't a 3-to-1, it was a 9-to-3 to 3-to-1. So 9 dominant round yellow, 3 round wrinkled, 3 wrinkled green, and 1 the recessive true breeding type. That would be green and wrinkled. Okay, and they came up with this 93 to 3-to-1 ratio. That's when doing a dihybrid cross. And what he learned from there is that the genes themselves assort independently. So because you had the thing for yellow and the thing for texture on different chromosomes, they were not associated with each other, and they were also at random. And we know this because we talked about this during meiosis, that the chromosomes align independent of each other. And that's called independent assortment. So different homologous chromosomes pair up during meiosis 1, or metaphase 1, and they go to opposite ends of the cell at random. Okay, so then the ability, if you want to determine the probability of getting one allele over the other, there are two rules. You have the rule of addition, which is if you have two mutually exclusive events occurring simultaneously, then that's the sum of the probabilities. So when you're crossing heterozygote purple plants, the probability of producing another heterozygote is the probability of obtaining one pp plus the probability of obtaining another pp. So a big P and a little p, or a big, here we have A's and B's, but the same concept. 2 or 1 out of 4 would be big B, little b, and 1 out of 4 would be little b, big B. Add those together, 1/4 plus 1/4 is 1/2. So the probability of getting a heterozygote would be 50%. The multiplication rule, again, if you have two independent events occurring simultaneously, the probability that they'd occur would be the product, or the multiplication of them. So you can see this in male and female, the probability of getting a male and a female. So females are XX, males are XY. The probability of obtaining a Y from the father, well, he has an X and a Y, so that would be 1/2. The probability of obtaining an X from the mother is 1. That's all she can donate. You multiply them together, and that's going to be your probability of getting XY, or a male, which is 1/2. And when you look at the world, this is the world map probabilities of males and females. Pink is females, blue is males, green is 50-50. Overall, it's about 50-50, except we mess with certain things with abortion and otherwise. So it doesn't quite, the probability is still 1/2, but we actually see something a little bit different because of other stuff. Alright, if you want to determine the genotype of a certain trait, one thing you can do is do a test cross, and Mendel did these. What he did was he did a recessive, homozygous recessive, and he paired that with whatever he was looking at, and based on the proportion of different individuals, you can determine whether it's what the genotype of it. Okay, and let's say if it was heterozygous, big P, little p, and you did it with a little, cross it with a little p, then you would get a 50-50 purple to white ratio. But if it was homozygous, dominant, and you crossed it with a homozygous recessive, then they would all be purple. So that's how you could tell. So Mendel did a very good job in these very simplified situations, but that's not how all genes work. His assumed that each trait was controlled by a single gene. Each gene only had 2 alleles, and there's a clear dominant recessive relationship between the alleles. But we don't see this. So, for example, these are my parents. This is my mom and my dad. They both have black hair, and they both have brown eyes. And so you would think, well, all of us would have black hair and brown eyes, or maybe black or blonde or brown and blue, but that's not the case. So we have brown hair, blonde hair, sandy blonde hair, dark hair. We even have a red hair. I have lots of kids in my family. A lot more than just two traits from one. And same thing with eye color. We have blue, we have green. Ooh, what's going on? Sorry. So Mendel does not hold true in every situation. For example, polygenic inheritance, poly meaning many, gene meaning genes. So polygenic inheritance happens when there is more than one gene controlling the phenotype or the expression of a trait. These show continuous variation, so rather than just being short or tall, you can be any variation between short, medium sized, average size, above average, and to very, very tall. And this is called a normal distribution. Pleiotropy is where one gene will affect multiple areas, and the example of that is cystic fibrosis or sickle cell anemia. Here's cystic fibrosis. All these conditions are caused by one gene. The heart, the spleen, the stomach has multiple effects because it's pleiotropic. You may also have multiple alleles, so there may be more than two alleles for a gene in a population. For example, blood types. You have A, B, or O blood types. So that's 3 different alleles, and we only have 2 available as humans. So this makes for multiple possibilities of different blood types. You may also have incomplete dominance, and this is more like the blended theory that originally was thought. Some things are blended. For example, these red flowers and white flowers, when you cross them, you get pink flowers rather than red or white when they are heterozygous. You also get red or white, but the in-between, it's called incomplete dominance. And then you also have codominance, where both of them are expressed. So this also happens in blood types. If your mom has A and your dad has B, and you get one of those alleles, then you will have type AB blood. Both A and B genotypes are expressed. Another one is environmental influence. So an example of this is Himalayan rabbits and Siamese cats. Different proteins or different melanins will be expressed based on the temperature of the cat. So different areas of the body, the nose and the ears and the tail, are cooler than the rest of the body, so they express black, and then the warmer parts of the body express white. Another thing is called epistasis, and that's where you have to have 2 functional products of the gene. So gene A produces something that affects gene B. Okay, and so in order to get purple corn, you have to have a big A and a big B. If you have a big A and little b, well, that doesn't work. If you have little a and big B, well, that doesn't work either. So you have to have both functional genes in order for it to work, and it leads to a very different proportion. Rather than 9 to 3 to 3 to 1, you have 9 to 7, or 3 to 1. Alright, so now we're going to talk a little bit about genetic material and how it's replicated. So first, genes, there's a big mystery as to how things were being, we know that there were genes and things were being passed from one generation to another, but we didn't know how. And the first real experiment which proved that there was something going on was done by Frederick Griffith. And what he did was he inoculated different mice with different strains of pneumonia, one of which was very deadly and would kill them, and it had a coat on it, polysaccharide coat. The other one did not have a coat, and when you infected a mouse with that, it lived just fine. But if you heat blasted the pneumonia and then put it in the mouse, it would also live. Now the kicker experiment, though, was when you combined these two, a heat blasted one and the non-deadly one, and you put them in a mouse, then it died. So the conclusion was, well, sorry, go back. Conclusion was there must be something going on, some sort of transfer of information between the 2 different types. Okay, so this experiment was repeated